of the day that's termed Scientific and Regulatory Uses of IVPT Studies. And of course, we all realize that many of us have used the IVPT in a research setting or R&D, perhaps in industry. But today we're going to try and bring the world of, the science lab to the regulatory setting and try and merge these and discuss how both of these worlds come together. And we'll start this session with presentations, and our first presenter is Dr. Yang, who is staff fellow at the FDA, and she's going to talk to us about IVPT studies with sunscreen products and the experimental parameters.
Thanks for the kind introduction. Good morning, everyone. So based on the method development principles that Sam just talked about, I'd like to present a case study of sunscreen IVPT, focusing on experimental parameters that are commonly considered in studying thermal absorption of both over-the-counter drug products and also the generic drugs. So I have a disclaimer here that this presentation reflects the views of the authors and should not be construed to represent FDA's views or policies. So sunscreen products are regulated as cosmetics in many countries, but in the US, most of them are regulated under the OTC monograph system. Therefore, we expect the quality of sunscreens consistently meets the expectations of the user. So in this session, I will talk about multiple experimental parameters to consider in sunscreen IVPT method developments. Sunscreen products contains UV filters as active ingredients to prevent sunburn and skin damage following UV exposure. Although sunscreens are supposed to act locally on the skin surface following thermal application, many UV filters have been detected in systemic circulations, which cause safety concerns. So we aim to develop IVPT for sunscreens to first support sunscreen selection in the maximum usage clinical trials or mass studies, or support the evaluation of frequently reformulated sunscreens. And also, if the method is successful, it may be also applicable to evaluate generic versus brand topical drug products. So this table summarizes active ingredients in all sunscreen formulations we tested in the FDA in vitro and clinical studies. The selection of sunscreen formulations for our study was based on the maximum allowable concentration in the monograph. For example, avobenzone at 3%, and other commonly used UV filters in commercial sunscreens were also studied. This table shows you the UV filters that we studied, and they all have distinct physical chemical properties, including hydrophobicity, molecular weight, melting point, etc. And their hydrophobicity, as indicated by the log P values, ranges from 1.4 to 6.8, and molecular weight ranges from 200 to almost 600 Dalton, indicating that some of them are more skin permeable than the others. So due to the large spectrum of physical chemical properties of these UV filters, a sensitive and robust analytical method is needed to analyze the permeation of all UV filters in an efficient way. All UV filters should be detectable and quantifiable, and instead of full volume removal and replacement for IVPT sampling, we performed aliquot sampling of 500 microliter at each time point to make sure that the receiver solution always has a suitable concentration of all UV filters for analysis. To ensure that the UV filters are fully dissolved in the receiver solution, we added BSA in the receiver solution as solubilizer. However, all UV filters should be extracted from the BSA solution before mass spec analysis. Therefore, sample processing method needs to be fully validated to avoid introducing errors. This includes during the BSA crash out or protein precipitation process, extraction of active ingredients, and sample dilution or concentration process, and also the sample stability should be studied for the duration of shelf life. So when selecting a diffusion device for IVPT, there are multiple options, including wet heat and dry heat systems. We have used French diffusion cells and flow-through diffusion cells from Permagear with wet heat system. The temperature of this system was controlled with water jackets and water heater or circulator. So our study used the traditional vertical French diffusion cell instead of the flow-through diffusion cell because the low receiver solution volume in the static French cell can ensure that all UV filters, especially those with slow absorbing ones with high molecular weight and high log P values, always have detectable concentrations in the receiver solution. So as shown in the picture on the left, full removal and replacement of receiver solution requires long plastic tubings. So to avoid non-specific binding of the UV filters to the plastic tubing, we use the Analytical sampling method instead of using these long plastic tubings. So the drawback of this system is that it's inconvenient to manually take samples throughout the night, whereas an auto sampler can do it without complaint. By way of contrast, the flow-through system has auto sampling function. Sorry, here. And it has a 6+1, so like a number 7 diffusion cell with one that can be used for blank control. And the sampling of this system is full removal replacement without introducing human errors. And for this reason, however, the sample volume can be much higher, and too much dilution of the sunscreen UV filters in the samples is unwanted. So there are also dry heat devices for IVPT. So far, we have tested the Diffusion Master System from Teledyne Hansen. It has an auto sampling arm as well, so provides tremendous convenience for overnight sampling. So the solution temperature of the Diffusion Master was controlled with heating blocks. And different from the previous Hansen system, the retired Phoenix system, the auto sampling of the receiver solution does not cause pressure change in the cells or at the skin surface, which protects the skin, especially when we use the very thin skin samples. So for example, human epidermis, it will prevent the skin samples from bulging or breaking. And we're currently testing sunscreen products with this system, and hopefully to share some more results next time. A similar design was found in Logan instruments. From the information of this instrument, we know that it has auto sampling function and also a tilting mechanism for air bubble removal. And it might be also convenient for full removal and replacement of receiver solutions, and as well as Analytical sampling. So the long sampling tubings shown in the picture might be a concern for non-specific binding with the UV filters. However, we haven't tried it yet. To select a suitable model for sunscreen IVPT, studies were carried out to compare permeation profiles using different skin models. To ensure permeation can be observed, only dermatoned human cadaver skin with an average thickness of 250 micron and heat-separated human epidermis with average heat thickness of 100 micron were used. So the advantage of using dermatoned human skin samples, including that the skin integrity can be maintained for a long time, as long as the bacterial growth is well inhibited. Also, both the infinite and finite dosing can be used as the integrity of the dermatome skin can be easily maintained. However, dermatome skin manually collected from human cadavers may have intradonor variations in thickness, and also interdonor variation of the dermal layer compositions could also contribute to data variations. So due to sampling is manual, using the vertical French cells, we were unable to collect permeation samples beyond 10 hours, as you can see from the graph at the bottom. However, although we observed a drastic change in the cumulative permeation between the 10 and 24 hours, So we did not observe very significant change after 8 hours in the flux curve shown on the right at the bottom. So this data justified our sampling schedule of not including late night samplings, which means most of the drastic change of flux happened before 8 hours, between 2 to 6 hours. So we also observed that both the heat-separated epidermis shown on the left side of this page and the dermatome skin model data shown on the right side of this page have demonstrated the same rank order of in vitro skin permeation of sunscreen UV filters. For example, here we've shown that oxybenzone from the A lotion showing blue curve and the B lotion showing red curve has similar or comparable oxybenzone permeation and the flux profile, but the B spray showing gray has significantly lower permeation than the two lotions that we tested in both skin models. So the results provided justification of choosing the HSE or heat-separated epidermis as more relevant skin model for sunscreen IVPT, especially we know that UV filters typically got absorbed to the epidermal layer and protect the skin from the basal layer of the epidermis. As compared to the dermatome skin, procuring HSE may introduce less variation in skin thickness, and HSE are good for short-term IVPT, such as the sunscreen study. Only 6 to 24 hours is required, so that the skin integrity can be maintained without worrying about overhydration of the epidermis. If overhydration happens, we can see it clearly from the cumulative permeation curve that a sudden change in slope will be observed if the overhydration happens. So in addition, we observed that infinite dosing may not be compatible with the HSE model. From the graph on the top that comparing the finite dosing group showing red with the infinite dosing group showing blue, that you can see the infinite dosing group has a much greater standard deviation at 24 hours as compared to the finite dose group, which is the percent CV is 22.5 as compared to the 9.3 in the infinite dose group. So we found that the amount of oxybenzone in the receiver solution, like when we calculate it for troubleshooting, it's less than 5 times of the oxybenzone solubility. So it means that indicating that in the skin condition might be compromised when we use infinite dose to the HSE skin model. And if we use a finite dose for IVPT with HSE, we can see that at the bottom panel, all the products has a permeation concentration at the most at the 24 hours that is still lower than its skin condition if calculated by 5 times of its solubility. So therefore, HSE may be only good for finite dosing, and it is also important to know that to obtain reliable data, sufficient HSE skin samples need to be obtained from multiple donors, and each donor should have at least 6 to 9 pieces of the good quality sample to allow for at least 2 to 3 repeat experiments for us to get the data. And we also performed a comparative study using HSE with infinite dosing compared with finite dosing in different sunscreen products. For example, here is A lotion versus B spray. So cumulative permeation from the infinite dosing group is always lower than that from the finite dosing group is always lower than from the infinite dosing group. And the flux max showed up mostly around 2 hours. So this fits the dosing schedule recommended in the sunscreen product label, saying that reapplying sunscreen every 2 hours to reach maximum sun protection. Although systemic absorption that we later on observed is definitely unwanted. So according to the FDA guidance for a cyclotocin topical cream, the cumulative permeation of the active ingredient for the duration of test would correspond to the clinical area under the concentration time curve, or the AUC curve. So we attempted to apply this concept to construct in vitro in vivo correlation between the sunscreen IVPT and clinical data. The original AUC data was obtained from the published FDA sunscreen clinical study in JAMA in 2020. And we can see that from the top table, there are 4 UV filters exhibited rank order correlation between the in vitro cumulative permeation with the clinical AUC. This is a level C IVIVC comparing rank orders only. And however, there are 2 UV filters, including octocrylene and avobenzone, shown on the bottom table, that does not have a very good correlations with between the 2 type of data. So the results indicate that the discriminatory power of this IVPT method may be higher for molecules with moderate to high skin permeation potential, shown on the top table, and for molecules with low skin permeation potential, like octocrylene and avobenzone, some more method development improvements should be is needed. So in summary, from our experience in developing IVPT methods for sunscreens, we want to emphasize that a fit-for-purpose IVPT method needs a lot of fine-tuning of the IVPT parameters. Either a well-accepted traditional method or a controversial method or novel method is used, a sufficient data comparison and justification is definitely needed to demonstrate that the method is applicable to the study. After all, comparing the rate and extent of drug absorption of permeation among different topical products is our ultimate goal of IVPT method development. So through this study, we found that both dermatone and HSE may be employed in sunscreen IVPT, and the rank order results of cumulative in vitro permeation may be correlated with clinical AUC. In addition, skin permeation of UV filters is influenced by their physical-chemical properties. Absorption of UV filters also depends on the properties of the skin sunscreen formulation, including excipients. So IVPT method may be extremely useful for comparing reformulated sunscreen against the sunscreens with clinical proven safety. So these are the references that supports our study. And I would like to acknowledge FDA funding support for this project, and also our group member from OPQ, Anne-Marie, Jiang, Daniel, Huzefi, and our management. And then also our collaborators and consultants within CEDAR in the working group, like Sam, Priyanka, and Dennis, and also collaborators from OND, and also this workshop planning committee for your feedbacks and invitation. Thank you.